Okay, good morning. We're going to go ahead and get started now. Does anybody have any questions on anything before we start the lecture? Okay. Then, if I remember correctly, and I went back and checked the video, we are still in cells, obviously. And I think we had just finished up the section on the glycocalyx structure. And correct me if I'm wrong in that, because that's what my recording, that's what the video shows. So we're going to start talking about, I'm assuming everybody can see this. It is a Monday morning and there are issues already with D2L, but it shouldn't affect us. Anyway, so I have us starting here on cell junctions. Now, cell junctions are how cells are held together. Cells are held together three different ways. We can have what are known as tight junctions, we can have desmosomes, and we can have gap junctions. And not every cell is tied into another cell to make tissues. White cells roam free through our plasma looking for things to kill. That's really simplified to answer, but that's what they do. Red cells float through our plasma carrying oxygen. Platelets roam through our plasma. Sperm cells and egg cells are not attached to any other cell. And so a lot of there are a fair number of cells that are not attached, and there are many, many more that are attached to each other. Now, three different ways. The tight junctions, almost got desmosomes and tight junctions mixed up there. Tight junctions are where we want to have a barrier where nothing can get through that barrier. So, for example, in our stomach, the lining of our stomach, the cells in the lining of our stomach, the epithelial cells that make it up are held together with tight junctions. Now, tight junctions look like they're sewn together. They look sort of like this, like they're stitching between cells, and they are impermeable. Nothing can get through there, and that's good because the contents of the stomach are very acidic. You know, when we're busy churning out hydrochloric acid, we don't want that to get out into the abdominal cavity. So we prevent the stomach acids and the stomach contents from leaking out. And hang on one second, let me get them. Okay, now. So we don't want stuff leaking out. They're held together. They're almost like they're stitched together, like clothing, or like in a tarp or something like that, or a tent. You know, how the pieces of a tent are stitched together. A totally impermeable structure. Nothing gets through, and that's to protect us from the contents of the stomach. Now, the second type of structure are the desmosomes. And the desmosomes are a little different. The desmosomes are very powerful. They're held together with what looks like rivets. We have keratin, waterproofing protein keratin that is strung between the walls of the membranes of the cells. And it's like they're anchored in, like they're bolted in place. So they give you great strength, but these keratin fibers that hold the membranes together are also very flexible. So we find these particularly in the heart. At those intercalated discs you saw in the lab, because the heart has to contract every 7/10 of a second. And it isn't some cells that have to contract. Every muscle cell in the heart contracts together. So it functions as one great big giant cell, about the size of your fist. And so it will contract at the same time with the same strength always. So we want to have that flexibility, but we also want to have great strength. We want to hold those cells together. So we hold the cells together with desmosomes. And we have We have the great strength of being held together, but the keratin fibers that run between them give. They're flexible because when the heart contracts, every muscle cell in the heart has to contract at the same time. So we have great strength there. Now the third type are the gap junctions. So the gap junctions are literally what they sound like. There are holes in the membrane that allow cells to connect to each other. They're like little passageways that go through here, or tunnels. And what will happen is these gap junctions will open from one side of one cell to another cell, and ions can pass through there. Electrical charges can pass through there. And so it's very useful for the rapid spread of a nerve impulse, for example, for our heart. Again, back to our heart. For contraction of our heart muscle, we want to have the electrical charge that goes through our heart muscle to go through as rapidly as it can. And so instead of having to come through the top of the cells and work their way down, the heart shoots its message through the inside of the cells through these gap junctions. So it's a very rapid way of communicating. So the cells hold together like that to form these tissues, whether it's muscle tissue or nerve tissue, whatever we're talking about. We have these three junctions holding holding cells together. Sometimes it's a combination of all three. But even though the cells are held together very strongly or have these openings in there, we still have to have ways to get materials into the cell and out of the cell. And the cell loves passive methods. The cell doesn't want to spend any energy. Energy is the cell's money. It's the cell's currency. And so the cell doesn't want to spend any more than it has to, and it prefers free. It prefers free transport. And of course, the best free transport is going to be diffusion, followed by, or filtration. Now, filtration only shows up in a couple of places in our bodies. We see it in our kidneys. We're filtering our blood 60 times a day through capillaries in our kidneys. We also have openings in all of our capillary walls. Our blood vessels are not a sealed tube. They leak. They have lots of openings. And so even at that level, we are filtering the larger molecules from going out of the plasma out into interstitial fluid. We contain it inside the cells. That's one type of transport. It's free. But the one that affects us most is diffusion. Simple diffusion, carrier-mediated diffusion, or osmosis. Osmosis is a subset of diffusion. Now, simple diffusion is the easiest. We go across a cell membrane from a high concentration of a solute to a low concentration, whether it's a liquid, another liquid, a gas, a solid, whatever it is. One side of the membrane has a higher concentration than the other, and substances always go down their concentration gradient freely from the high concentration to the low concentration. The molecules are moving around on their own. They're all moving around, and one side of the membrane has lots of molecules and one side has few molecules, and molecules will move randomly down across the membrane to reach equilibrium. This is simple diffusion. This is the concentration gradient. We go from the high concentration to the low concentration until we reach equilibrium. And there is no energy required for this to happen. The molecules are already moving randomly. If a solute is in water, the solvent is water and it's a solute, the solute particles will randomly move on their own. The water molecules are randomly moving on their own. We don't have to do anything special to get the solute to go across the membrane, or whatever the high concentration side is. Now, simple diffusion affected by the size of the particle. Little particles diffuse faster than big ones do. So large molecules have a problem. diffusing. Temperature plays an important role. Temperature, the greater the temperature, the faster the particles are going to move, and the quicker diffusion is going to reach equilibrium. So diffusion, particle size, and temperature are going to play a major role here. It's going to be harder for substances to diffuse at colder temperatures than at warmer temperatures. Now, the problem we have, though, is the plasma membrane. The plasma membrane is selectively permeable or semi-permeable. It doesn't let things through that are water soluble. It loves fat. It loves fat soluble products like lipids or substances suspended in lipids like the fat soluble vitamins or the steroid hormones or cholesterol. Those things have no trouble diffusing across the membrane. Very tiny molecules can diffuse across the membrane. Oxygen, CO2 have no trouble. They can slide across the membrane. But the membrane stops most other things. It is a barrier. It's a barrier to the movement of ions. It's a barrier to the movement of water. It's a barrier to the movement of glucose and proteins and hormones and anything else that wants to get into the cell cannot just slide across the membrane. The membrane is a barrier to simple diffusion. So the only things that can get through that membrane is anything that is fat soluble. Anything that's fat soluble, anything that is non-polar, meaning water's left out. Gases can get through because they're extremely tiny. Gas molecules, O2, atmospheric oxygen, 2 oxygen atoms linked together, it's pretty tiny. Same with CO2, it's a pretty small molecule too. So there are no issues with the gases going across the membranes. If we have larger molecules or if we have ions that we need, like sodium and potassium and glucose, sugars, we have to have special proteins called carrier molecules that can get them through the membrane. Because we need them. We need glucose. We need glucose, we need sodium, we need potassium. We have to get them through the membrane. The water-soluble vitamins, we have to get them through the membrane. So we have to have workarounds to let them through. Problem with the plasma membrane is that if it's damaged, then the contents of the cell will leak out. A burn patient that is burned with third-degree burns, and that's down to the bone. For a really graphic image, think of barbecue. Barbecue on the grill, cooked, done. If you have barbecued ribs, that's what, and it's cooked all the way through. That's what third-degree burns look like. Burned, the skin's burned off, you're burned down to the meat, you're burned down to the bone. And the cells that have survived that are going to be damaged, and they're going to leak. Fluids leak out of damaged cells tremendously. One of the biggest problems we have with severe burn patients is that not pain, because most of the burn pain receptors have been burned away, but is leakage, fluid loss. As fast as the fluid comes into their body, it's going to leak out through these damaged cells. And they lose water, they lose ions, they lose proteins, and it sort of oozes out. On top of that, it creates a great area for bacterial growth. So your patient that has severe burns, you're dealing with fluid loss, you're dealing with tissue damage, you're dealing with shock, you're dealing with loss of ions and proteins, and you're dealing with bacterial infection possibilities. So there's a lot of work going on there to support that patient. With simple diffusion, high concentration to low concentration, it works pretty well. Oxygen diffuses into our cells from our blood through simple diffusion. Oxygen concentrations in our blood are always higher than in the cells, so oxygen will diffuse across the membrane. Carbon dioxide is always higher in the cell than in the blood, so carbon dioxide diffuses out. And it works. That simple diffusion process works very well for us. When we don't have simple diffusion, we have what's known as facilitated diffusion, where we have a channel protein or a carrier protein, one of these integral proteins embedded in the cell membrane that will allow ions to come through. Sodium that we want to get out of the cell or into the cell, potassium that wants to get out or into the cell, glucose that we want to get into the cell. Something that is too big or is a polar molecule, or or is an ion or a vitamin or a protein that we want to get in. Now, a carrier protein or a channel protein. So a channel protein can just be a tube. Water usually comes into the cell through channel proteins. We call them aquaporins, like a water tube, or a hose, if you will. So it bypasses the phospholipid barrier. It just goes right through it. Water can go through there. Carrier proteins are a little more specific. Carrier proteins, and neither one of these channel or carrier proteins use energy. But the carrier proteins are specific, usually specific for different types of ions. For example, in this illustration, this animation down here, we see a carrier protein that opens up on one side and lets a substance come through. Now that may be an ion, maybe a specific type of ion. It may be glucose. It's how we get substances into the cell passively, bypassing the membrane. And it's the role of insulin in our cells. Insulin, we release insulin when our blood sugar levels go up. Insulin is thought to stimulate the production of carrier proteins in the cell so that the cell can take in the sugar, the glucose, if you will. So, and that's I think how we think. So I said that twice. That's how insulin works in the cell membrane, by stimulating production of these carrier proteins. And they're very selective. They won't, something that lets in sodium will usually not let in potassium, or something that lets in glucose is not going to let in a protein, for example. So this is facilitated diffusion, diffusion with help. You're still going down the concentration gradient, but now we found a way to get through the barrier. So again, these are integral proteins. They can take sugars across, they can take amino acids across. The carrier protein molecule simply changes its shape when the substance that we want to get across the membrane lands on it. There's no energy involved. Just when it lands, when like glucose lands on its carrier protein, the carrier protein will change its shape and allow the glucose to come into the cell, or sodium, or potassium. And again, they're usually very specific as to what they let through. These carrier proteins will only let the substance that they're designed for, and nothing else can come in with it. Now, there are some examples, but we'll get to that in a minute. Now, the channels are simpler. The channels are just tubes. Here we see a channel in the animation for sodium. For potassium to go out of the cell. It's leaving the cell. And this is how we allow how osmosis works. And the movement of water across the membrane uses this channel called an aquaporin to allow water to go into the cell or out of the cell. Again, based on the concentration gradient of the water. Now, what we call the leakage channels are always open. What you're seeing here is a leakage channel here, and that works. Now, there are other channels that have gates. They have gates that only open and shut at certain times. Now, some of the gates are controlled by electrical signals in our bodies, and some of them are controlled by chemical signals. The gated channels are how we control the movement of ions in our muscle cells and in our nerve cells. And we'll get into that when we get start getting into chapter 9 on muscle cells. But right now, all we have to look at is the leakage channels that let things move across the membrane. It's passive. It's based on the water concentration. These leakage channels for water are based on the concentration of the water on either side of the membrane. These passive processes are free. These passive processes are free. There is no energy spent to get them to work. However, there are times when we have to spend energy, and that's what we call the active process or active transport. We're moving something across the membrane one way or another, either into the cell or out of the cell, but we're going against the concentration gradient. Everything up to now has been we go from a high concentration to a low concentration. What happens when we want to go from a low concentration to a high concentration? We're pumping something out of the cell. We're pumping against the concentration gradient. We're pushing it out. It's like we're pushing something uphill. So, riding a bicycle, going downhill is easy. It's free. You don't have to do anything until you get to the uphill side of a hill, and then you have to pedal and work hard to spend energy to get up the hill. One side's free, one side isn't. Now, passive is great, but a cell sometimes has to get stuff into it or out of it against a concentration gradient, and then that's where it spends energy. It spends energy in the form of ATP, and we call that active transport. Now, the third type of passive processes is osmosis. Osmosis, we talk a lot about osmosis. Osmosis is the movement of water across a membrane from a region of high water to low water. What do I mean by high water or low water? Well, what is the percent solute concentration on one side of a membrane? If you have a cell in a beaker and the concentration, and you have a fluid in a beaker and you have a cell sitting inside the beaker in the fluid, the concentration of the solution in the beaker is 10% sodium chloride. Okay, so it's 10% salt and 90% water, because 10 and 90 make 100%. Okay, so 10% salt solution is 90% water. The concentration inside the cell is going to be 1% sodium chloride and 99% water. So one side of the cell, the fluid in the beaker has 10% salt solution, which means it's 90% water. The fluid inside the cell is 1% salt, which means it's 99% water. Now, we have the membrane separating the contents of the beaker with the contents of the cell. We have the cell membrane separating those two. The inside of the cell at 99% water has more water than outside the cell, where it's only 90% water. So water will leave that cell. Water is driven across the membrane from the high concentration, high water side to the low water side, whether it's 10%, whether it's 90% water, 60% water, 40% water, 80% water, whichever side of the membrane has the higher water concentrations is where the water is going to come from. Our cells are normally 0.9% sodium chloride. The interstitial fluid is 0.9% sodium chloride. But as the water level rises outside the cell and the salt concentration goes from 0.9% to 0.8%, that means there's more water outside. Water will come in. Water will go through these aquaporins. Now, fortunately for us, the water concentration inside and outside the cells stays about the same. It stays about 0.9% sodium chloride, which means it's 99.1% water, so we don't get any net movement of water. But if there was a change on one side or the other, that would drive the movement of water. Consider this. If you have a case of bottled water and you start chugging the water, I don't know why, but let's just say you start chugging the water down as fast as you can, and you're into your 7th or 8th bottle. Now, where's all that water going to go? Well, when you drink that water, it goes into your stomach, and from there it's absorbed in the small intestine, goes to the liver, and then it goes from the liver into your blood. So everything you eat and everything you drink goes into your blood. It's in your plasma. The water now is in the plasma. Where's plasma go? Well, plasma leaks out into the interstitial spaces. So now what you've done by drinking 10 liters of water, which is pretty hard, but you drink 2 or 3. Well, maybe you do drink 4 or 5 liters of water all at once, one after another. I mean, it's possible. So you have all this water now, and now it's flooding the interstitial spaces. It's diluting the salt concentration out there. You're no longer 0.9% sodium chloride, you're 0.8%, 0.7%, 0.6%, because you're adding more water to the salt that's there. What's going to happen to that water? It's going to move through osmosis into the cells, and the cells are going to start swelling up. And cells don't like that. Cells don't like to swell up, particularly, whether they're in the blood or whether they're tissue cells. Cells in the central nervous system do not like to swell. They don't like that at all. It can cause permanent damage if we become overhydrated. There's a term called water intoxication that can happen where we drink too much water. So water, if we flood the interstitial spaces with water, that water is going to go inside the cells because there's less water inside the cells. If our water concentration, if the salt concentration outside the cell drops to 0.7 and the salt concentration inside the cell is 0.9, we have more water outside. It goes into the cells. If, on the other hand, we become dehydrated and we lose water, our salt concentration goes up. See, we don't ever lose the salt. The amount of salt stays the same. Its concentration varies by how much water is there. The salt's not going to go anywhere. Imagine if you have a beaker full of water, and I've probably given you this example before, but if you have a beaker full of water, you have a liter of water, 1,000 milliliters, and you make a 10% salt solution out of that. So it's 10% salt, and you set it aside. And because you put 100 grams into a liter of water. Now, when the water will evaporate. When the water evaporates down to the 500 milliliter mark, half the water is gone. The 100 grams of salt are still in that beaker. The water now, the solution is no longer 10% salt, it's 20% salt, because 100 is 20% of 500 mils. And the more water that evaporates, the saltier that solution gets until all the water's gone and the 100 grams of salt are left behind, because the salt doesn't go anywhere. This is what's happening in our bodies. If we lose water in the interstitial fluid, if we lose plasma, if we become dehydrated, we get saltier outside the cells. What happens to the cells? they shrink, they give up their water. All of us, when you're dehydrated, the cells have more water than the interstitial fluid does. So water leaves the cells and the cells shrink. They don't like that either. So, okay. Now, this summarizes what I've just been talking about. The term osmolarity measures the concentration of the particles of solute in the solution. Yeah, so a 10% salt solution is 90% water. A 40% salt solution is 60% water. If you have 90% water on one side of a membrane and 60% water on the other side, water is going to go from the 90% side to the 60%. It doesn't matter what the particles, what the solute is. It can be glucose on one side and sodium chloride on the other side, but they're going to be driven by the amount of water. The way to remember it is that water always goes from the high water side to the low water side, because high water equals low solute. And so low solute is high water and high solute is low water. So it doesn't matter what the solute is. It's all driven by the amount of water that's present. So. And the cells like to reach equilibrium. They like to be in equilibrium with the inside of the cell wants to be in equilibrium with the outside. And however we have to shift water around to do that, the cell will. Whether it brings water in from the outside in an attempt to reach equilibrium, or it sends water out in an attempt to reach equilibrium. Water inside the cell is pushing out, and osmotic pressure is pushing the water in.
The plasma membrane is permeable to water molecules.
Okay, so we are not plant cells. We don't have a cell wall. We do have a cell membrane. And the cell membrane doesn't give us the protection that the plant does, but we don't have to deal with the inflexibility of a cell wall. However, we can see, the cells very easily move water back and forth, and we can have issues. Overhydration, underhydration can occur. Sometimes, well, I will say, whether a person is overhydrated or underhydrated can have fatal consequences. Now, a cell can only deal with so much water. It can only deal with so much water loss or water gain. The terms here, hypertonic, isotonic, and hypotonic, refer to the concentration of the solute outside of a cell. What's in the interstitial fluid? In a hypertonic environment, the solute concentration is higher than the cell. So water will leave the cell, because if we were to take a living red blood cell and place it in a 10% salt solution, which is 10 times greater than what the cell's used to, maybe 11 times, whatever. It's 10 times greater than normal for the cell. The cell will respond by sending water out of the cell contents, and it will shrivel up. It will crenate. You put a red blood cell in a 10% salt solution, and it will crenate. It will shrivel. If you put a red blood cell in a isotonic solution, you place it in 0.9% sodium chloride, which is the same contents as inside the red blood cell, nothing's going to happen. Water movement in and out of the cell is balanced. If you were to place a red blood cell in distilled water, 100% water is going to rush into that cell, and it's going to explode, and it's going to explode very rapidly. We used to demonstrate this in lab with red blood cells, and we put a drop of blood on a microscope slide, project it up on the screen, and then add some distilled water to the side. And the red cells, which were highly visible before, just turned into a red smear because they exploded. They lysed, they ruptured. It's like blowing up a balloon too much. So the cell membrane can't handle it, and it explodes, and that is irreparable damage. Now, if a cell can lyse, a cell can crenate. Cells can come back from being crenated, but when they shrivel up, they can rehydrate themselves as the water level becomes balanced. They can't recover from lysing. There we go. So this summarizes it. Isotonic solutions, the osmolarity, the concentration is the same inside the cell as outside. Any water movement in is balanced by water movement out. There is no net change. A hypertonic solution has a greater osmolarity. The solute concentration is greater. Water is drawn out of the cell. The cell will crenate. Hypotonic solutions, the osmolarity is lower. So water will go from the higher water side into the cell, and the cell will swell up and lyse. And this is pretty important. This is one of the reasons why we don't administer distilled water to patients in an IV line. IV line, you have a patient has an IV line going there. It's usually called normal saline. It's an isotonic solution that matches the plasma, matches the concentration of the interstitial fluid, matches the concentration inside the cell. The osmolarity is always the same in here. If you gave a patient distilled water, then blood cells would start rupturing because distilled water would rush into the red blood cells and they would start to explode, as well as the white cells. The only time that you would administer something other than isotonic solutions to a patient is if they are swelling up. They've been stung by a bee and they're having an allergic reaction, or they have congestive heart failure. And in those conditions, they are experiencing edema where the cells are swelling. Water is going into the cells. In congestive heart failure, there is so much, the fluid return back to the heart is not equal on both sides. So fluid backs up and water moves into the tissues. And in the case of an allergic reaction, the bee venom, for example, causes the release of a substance known as histamine. You've heard of antihistamines. Well, histamine is released by our cells and that histamine causes our capillaries to become even more porous, allowing more fluids to leak out into the interstitial spaces. We swell up. If we have a systemic reaction to a bee sting, we're going to swell up. We're swelling because all that extra fluid from our blood's going into the cells. A hypertonic solution would draw that water back out of the cells. So anyway, this is why it's very important that we maintain, we want to maintain osmolarity to be the same on either side of the membrane. Now, let's try to get this to, there we go. Okay, so that is diffusion, facilitated diffusion, and osmosis. All three are passive processes. Even facilitated diffusion is a passive process. We're simply moving down the concentration gradient. Active transport is different. Active transport and vesicular transport are both different. They require the use of ATP. They're moving against the concentration gradient. Cells don't like that. It's essentially pushing something uphill. Active transport requires ATP to power a protein pump. It actually pumps a substance across the membrane. It has to pump it against its concentration gradient. It has to actively bring in something that's not lipid soluble or something that is just too big to fit through the channels. And the channel proteins, they're not going to come across that way. We have to actively go against our concentration. So we have pumps. We have we have pumps that are carrier proteins, specialized carrier proteins that will pump a substance across the membrane. There are two types. There's what's called an antiport and one called a symport. An antiport will bring one substance in while it's sending another one out, like a two-lane highway. One goes in, one goes out at the same time. Symports bring two substances in that are different at the same time. One side of the symport brings in one molecule, and the other side brings in a different molecule. They're both going into the cell or out of the cell. But a symport has everything going one way. It's taking two substances into or out of a cell. Antiports bring one in while taking one out. They go in opposite directions. Both of them require ATP for energy to power the pump. Now, active transport can be broken down into two types. We have primary active transport, which requires ATP. Secondary active transport is a free ride. Secondary active transport depends on the pump, the ATP powered pump, the protein pump. Secondary active transport depends on that pump to keep the concentration of a substance high on one side of the membrane, so it can leak back in. We pump it out, it leaks in. We pump it out, it leaks back in. That's secondary active transport. We pump sodiums out. We pump sodium out of the cells, and it leaks back in. We pump it out to where it's high on the outside of a cell, and it leaks back in. That's what it does. We pump potassium into the cell, and it leaks out. So we spend a lot of energy, 40% of a cell's energy. We spend a lot of energy pumping stuff across the membrane only for it to leak back in, and that is the secondary active transport. We don't have to spend any energy for it. We've already done that. But it leaks back in. You pump it out, it leaks back in. As far as primary active transport goes, we are using ATP to change the shape of the carrier protein embedded in a cell membrane. It causes the protein, the pump, to open from one side to the other and then close again, back and forth, back and forth. And we see these pumps, we have calcium pumps. We depend on calcium in our heart muscle. We have, when we get into AP2, we'll talk about what are medications known as calcium channel blockers, which we use to among other things, regulate blood pressure. Calcium channel blockers are very important in regulating blood pressure and also regulating the heart rate, because calcium is pumped across the membrane in the heart. Something else that we encounter a lot are proton pumps. Acid reflux is caused by high levels of acid in the stomach being regurgitated up into the esophagus and burning the walls of the esophagus. One way to control that is to take medications that shut down the hydrogen pumps, the proton pumps. These protons, these hydrogen ions, because that's all they are, is a proton. There's no electron in the hydrogen ion. The hydrogen ion, we know that acidity is driven by the presence of hydrogen ions. The more hydrogen ions you have, the more acidic you are. So what we have here is our medications like Prevacid and Protonics, for example, that are proton pumps that shut down these proton pumps that are creating the acidic conditions in our stomachs. So, because the pump itself is normally turning out all these hydrogen ions. We shut it down. We use what are they're known as these medications are called proton pump inhibitors, and they shut them down. Not all of them, but they shut down some of them. The other type of pump, the one that we know more about than any other kind, is the sodium potassium pump. More work's been done on the sodium potassium pump because of what it does. And the cell will spend about 40% of its budget, its energy budget, just on the sodium potassium pump alone. Now, the sodium potassium pump has to deal with the fact that there are always normally high levels of sodium outside of our cells. So the two most important ions in our bodies are sodium and potassium. Sodium is always high outside our cells. Potassium is always high inside the cells. Consequently, sodium is always going to be leaking into a cell. Sodium's high outside a cell, it will leak in. It will find a way to get in. It will leak in through a sodium channel. Potassium is always high inside the cell and it wants to leak out. So we have sodium leaking in and potassium leaking out. We need to maintain these concentrations. The ability to generate nerve impulses depends on the difference in the charge on the cell between the sodium and potassium. See, our cells are all charged. Muscle cells have a charge of minus 90 millivolts. Muscle cells have, nerve cells have a charge of minus 70. Muscle cells are minus 90. And that charge is based on the concentration difference between the sodiums and the potassiums. If the sodiums were to leak in continuously till they reached equilibrium, and the potassiums leaked out till they reached equilibrium, we'd have no charge and the cell wouldn't work. The cell would not be able to do its job. So what we do is when sodium leaks in, we pump it back out. When potassium leaks out, we pump it back in, and we do it at exactly the same time. The sodium potassium pump is an antiport that will pump sodiums out and potassiums in. And we always pump, and this is probably relevant only for a test question, and you need to remember this, we always pump 3 sodiums out and two potassiums back in. 3 sodiums out, 2 potassiums back in. And that heat maintains the charge on the membrane. It maintains at minus 90 millivolts on muscle cells and minus 70 millivolts on nerve cells. And we don't have that charge, we can't generate nerve impulses. We can't get muscle contractions. Cell's dead. Got to have that charge there. So when sodiums leak in, we pump them back out. We pump out 3 of them. We see what's happening here. There's the sodium potassium pump. In this state, it's open to the inside of the cell membrane. Sodiums drift in there. As soon as the 3 sodiums are in place, it's only designed to hold 3. ATP will come along conveniently because it's always available, and cause the sodium potassium pump to shut this side of the membrane, open the other side, let the sodiums leave. After the sodiums are gone, we see 2 potassiums come in, and their presence in the sodium potassium pump with 2 potassiums will cause the outside of the pump to shut and the inside to open, allow the potassiums to come in. We're ready for more sodiums. And it looks something like this. Little yellow circles are sodium, and the big green ones are potassium. I don't think that's how they're colored, but so the sodium goes into the pump when it's open to the inside, but it requires ATP to do something. And that will allow, when we have ATP present, it will push the sodiums out, and then it opens up for potassium to come in. If we run out of ATP, we can't get rid of the sodiums. The sodiums will continue to leak in, the potassiums will continue to leak out. When we run out of ATP, the cell dies. And so, because we're spending so much energy, 40% of the cell's energy budget is spent just on this. 3 sodiums out, 2 potassiums back in. Every cell has a sodium potassium pump or pumps embedded in their membrane. It's most important in muscle cells and nerve cells. So this slide summarizes everything you need to know about the sodium potassium pump. You need to know 3 sodiums out and 2 potassiums back in. Is that going to be a hard-hitting healthcare question later on? Not directly, but you have to understand why we need sodium, why we need potassium, because we have to have them in the cells to maintain this charge. So while the fact that the pump is pumping 3 sodiums out and 2 potassiums back in is a nice test question. Is it going to be relevant to something in healthcare? Well, I don't think my patient is pumping their sodiums out like they should. No, but sodium levels are important. Potassium levels are important. And how the cell maintains that is through the use of this pump. Come on. And so you see on the left-hand side here, we see the sodium potassium pump. It's not animated, but you can see it has 3 slots for sodium to go out and 2 slots for potassium to come back in. That's primary active transport. Secondary active transport is different. It's a free ride. Because we're maintaining the concentrations of sodium very highly outside the cell, when sodium leaks back in, it will often bring along glucose. We have what's known as the sodium glucose symport. Sodium glucose symport. This may be how insulin works, by creating this symport that allows sodium to come in at the same time glucose comes in. When the sodium lands on the symport, when sodium lands on the sodium glucose symport, it will allow the protein to change its shape to create a spot for glucose to land next to the sodium. And that cause when the glucose lands on the protein on the symport protein, it causes it to open up to the inside of the cell, and both sodium and glucose come in. Now the sodium is going to get pumped back out, but the sodium is leaking in and dragging along glucose with it. And this may be what insulin does for us by creating these specialized carrier proteins. This is a passive process on secondary active transport, but it does allow sodium to drag along the glucose in this symport example here. So another reason why sodium is important to us, it helps us bring in glucose. So now the other example of an energy, like an ATP energy requirement, an active type of transport is vesicular transport. ATP is required to physically engulf a substance and bring it into the cell, or to physically kick something out of the cell. We have what's known as endocytosis, where we bring in solids, that's phagocytosis, liquids, that's pinocytosis, or we have what's known as receptor mediated, meaning we only bring in certain things that fit receptor sites on the cell membrane. The top illustration there is a video of a white blood cell engulfing a bacteria. And you can see it coming up, the blue, okay, here it comes, and it engulfs that bacteria and takes it inside its own membrane, and it destroys it. That is phagocytosis. White cells look like an amoeba. They engulf a substance. They put it inside a little vesicle. A vesicle is a bubble that is made of the same structure as the cell membrane. It's a phospholipid bilayer. So when it surrounds it, it can surround it and pinch it off in its own little vesicle, and then it can bring it into the cell and destroy it. And it does that with liquids. It does that with specialized receptors too. Exocytosis, we've made something. We've made a protein. We want to release it from the cell. With exocytosis, we've got the protein inside a bubble, and it goes from inside, that bubble, that vesicle has to go fuse with the cell membrane, fuses with the cell membrane, and it gets released. It never allows the membrane to open to the outside. Never allows the membrane to open to the outside, but it does release the contents. We can see here in this bottom illustration, here comes a vesicle containing a protein. It fuses with the cell membrane. The vesicle opens up and allows the substances to go out. But the membrane never gets compromised. The integrity of the membrane is never broken. The contents of the inside, the cytoplasm is never exposed to the outside. Here we see endocytosis. We engulf something here in the left, large particles, cell fragments, worn out structures, worn out organelles from inside the cell. All these things are engulfed. A cell dies, and the all of its friends and neighbors eat it by engulfing it. Or a liquid with pinocytosis, or receptor-mediated endocytosis, where we only take in certain things. Exocytosis, the substance gets formed in its own and stored in its own vesicle, and then gets released. The vesicle fuses with the cell membrane. It is made of the same substance as the cell membrane, and then you see it right here, and on the right-hand side of the screen, we see the contents of a vesicle being released to the outside of the cell. So those are proteins or whatever that are being released. It's 100,000 magnification, so whatever it is it's pretty small. Now, so that covers the movement across the membrane. Now so the membrane is pretty specific. It's a phospholipid bilayer. It is designed to protect the cell. It's designed to control the movement of substances across the membrane. It's designed to keep fluid inside the cell and fluid outside the cell separate. So it has these specialized structures here. Now the cytoplasm, the cytosol, the cell sap, the stuff that's inside the cytoplasm. Used to, 70 years ago, it was called protoplasm. Then it became cytoplasm. Now it's considered cytosol. It's sort of like, may have the consistency of hotcake syrup, if you will. It's got proteins embedded in there. It's got salts and sugars and vitamins and hormones and enzymes and all sorts of things like that. It has glycogen, it has salt crystals, and it has organelles in the cytoplasm. So it's a very busy structure. The organelles are like little organs that are going to do things for the cell. So we have, and most of the organelles are surrounded by a membrane that is very similar or identical to the cell membrane. The phospholipid bilayer, mitochondria, the endoplasmic reticulum, the Golgi apparatus, peroxisomes, and lysosomes all have membranes. The ribosomes and the cytoskeleton and the centrioles don't. It's a hard-hitting factoid there. So, well, this is a mitochondria. Well, that is, we'll talk about that first. Mitochondria is the energy conversion facility in our cell. It takes the energy from glucose, oxidizes it, releases the energy, it releases its potential energy by breaking the covalent bonds in a glucose molecule. It doesn't make energy. It converts it from the potential energy in glucose to the potential energy in ATP. This makes a chemical change, or converts it from glucose to ATP. So we make all of our ATP, or most of our ATP, I should say, in the mitochondria, and we use that ATP to do things. Now, the problem with ATP is we only have a 7-second supply, so we're always making new ATP over and over and over again. As long as we have glucose, we can make ATP. As long as we have oxygen, well, that's not even true. We can make glucose, we can make ATP without oxygen. As long as we have oxygen, we can make a lot of ATP. As long as we have glucose, we can make ATP in general. The bulk of the ATP production, the bulk of the conversion of the energy in glucose to energy in ATP takes place in our mitochondria. They look like little sausage links. Now, the ribosomes don't have a membrane, but that's okay. They don't need a membrane. What they do, the ribosomes are used to assemble proteins. It's at the site of protein synthesis. The ribosomes are where amino acids are brought together and joined with peptide bonds so that they can form a protein. Because what's a protein? A protein is nothing but a chain of amino acids linked together with peptide bonds. So this chain of amino acids are assembled inside the ribosomes. That's what the ribosomes do. Most of our ribosomes are attached to this next structure we're going to see here, the endoplasmic reticulum. Some ribosomes float free. But most of the ribosomes are bound to the endoplasmic reticulum, which looks like this: a series of tubes which surround the nucleus. Now, the image here, you have to use a little bit of imagination. The endoplasmic reticulum completely surrounds the nucleus like a ball. The nucleus is a ball. You have a larger ball surrounding it. That ball, these tubes, these flattened tubes, are the endoplasmic reticulum. Now we see it in two varieties. We have what's known as the rough ER and the smooth ER. The rough ER is studded with ribosomes. This is where we make proteins, in the rough ER. Proteins are assembled in the ribosomes, which are bound to the rough endoplasmic reticulum. And from there, the proteins then are then repackaged and prepared to be sent out of the cell. The smooth ER surrounds the rough. The smooth ER has a variety of functions depending on the cell type. Doesn't have any ribosomes. It is continuous with the rough ER. Now, what do we do with the smooth ER? Well, it's very important for us. In our muscle cells, we use our smooth ER, the smooth endoplasmic reticulum, to store calcium. Lots and lots of calcium. We need calcium for our muscles to contract. And we will raid our bones as we age, at least particularly in your postmenopausal females, because there's no estrogen. Estrogen stimulates bone growth and bone deposition. Without estrogen, you don't get that. So particularly in your postmenopausal females who are no longer making estrogen, the body makes a distinct determination that it would rather have the calcium from the bones to allow muscle contractions than to have it in the bone. So we break down, we see a breakdown of bone over time. If it's significant, we call it osteoporosis. Now, so we store a lot of calcium. We all store calcium around our skeletal muscle cells in these smooth ER. We use our smooth ER to break down glycogen to glucose. We use it to make steroid-based hormones, and we use it, the smooth ER, to transport fats. And most importantly, is we use the smooth ER, save the best for last, to detoxify chemicals. So our liver, which is the big detoxifier in our bodies, the cells of the liver have vast amounts of smooth endoplasmic reticulum, because this is where we are busy detoxifying. We are breaking down the chemicals in the food we eat, whether they are pesticide residues in our foods, whether they are food additives, whether they are preservatives, whether they are coloring agents, food dyes, whatever they are, we break them down. We break down medications. We break down medications in our livers to what our cells can truly handle. Consider the adult dose of Tylenol. An individual takes 2 Tylenol tablets, 1,000 milligrams of Tylenol. What gets through the liver is something less than 1,000 milligrams. Tylenol only works by blocking pain signals. It doesn't know where to work. It just goes throughout the body. Whenever there's a pain signal, it will block it. Just like Advil works by reducing inflammation. It doesn't know where to work. It just wherever, it circulates through the body. Wherever we have inflammation, it reduces it. But the dosage of Tylenol or Advil or aspirin or anything else depends on what's going to get through the liver. The liver will detoxify the Tylenol or the Advil or the aspirin. And what percentage of that gets through is left to work in our bodies. The same for any kind of prescription medication, amoxicillin or tetracycline or anything like that. Any of the Cipro or any medication, whatever gets through. And that's one of the reasons why we have dose limits on medications. You know, a children's dose of Tylenol is a much smaller dosage than an adult dose because a children's liver is smaller. The systemic circulation in the child is smaller. You know, so their liver can't process that much Tylenol. You wouldn't give 1,000 milligrams of Tylenol to a child. You may not give 1,000 milligrams to someone, you know, an elderly person whose liver may not be functioning at 100% anymore because it's old. Again, the dosage may vary based on the ability of the liver to detoxify. You know, this happens. You know, this is what the liver does using its smooth ER. The liver does this. The liver breaks down alcohol. The liver breaks down you know, any kind of medication. It breaks down any kind of food additive that might not be, well, whether it's good for us or not, it'll break it down. You know, consider alcohol. You know, the liver can break down an ounce of ethyl alcohol an hour. It breaks down the alcohol. It converts it ultimately to carbon dioxide and water. And if you're drinking 1 12-ounce bottle of beer and you make it last for an hour, then you're not going to have any ill effects from that because the 1 ounce of alcohol that is in that 12-ounce bottle of beer is going to be broken down over the course of an hour by the liver. It will break it down. All the intermediate byproducts like the formaldehyde and the formalin and the methanol that forms breaks apart as fast as it's formed. And there are a lot of some really ugly intermediate chemicals that get formed in the breakdown of ethyl alcohol, but they themselves are broken down instantly. It's when you have a patient that exceeds that rate that we get problems. The liver can only do 1 ounce an hour. What if you do 2 ounces an hour, or 3 ounces an hour, or 4 ounces, or 6 ounces, or 10 ounces, 10 bottles in an hour, or maybe 10 drinks in an hour, or 10 glasses of wine in an hour? You're going to suffer the consequences. You're going to experience what we call a hangover. The hangover is caused by the liver not keeping up with the rate of alcohol consumption. It will eventually get to all of that. But what happens when your patient overindulges? The hangover is caused by the accumulation of these intermediate toxic chemicals that the liver is still working on. And so your patient's going to feel miserable or uncomfortable until that's gone. So that's what the smooth ER does. That's what, the liver is all about detoxification, and that's probably the biggest role outside of storing calcium for muscles. It's probably the biggest role that we see the smooth ER doing for us. So now another structure that we encounter is the Golgi apparatus. And I told you we make proteins in the rough ER. Well, the rough ER doesn't care what it makes. Each ribosome in there, and there are thousands and thousands of ribosomes. Each ribosome is making a different protein at the same time. They're following whatever instructions are coming out from the nucleus. The DNA sends instructions out to the ribosome. Here, make this. We need more hemoglobin. We need more hair color. We need more eye color. We need more pigmentation in our skin. We need some of these enzymes. We need whatever. And so these ribosomes are busy chugging out proteins. Well, these, as they're formed, they're collected in a vesicle. And you have a whole variety of proteins in the vesicle. We need to sort them out so that we have all, all the hair color, all the eye color, all the skin color, all the hemoglobin, all the enzymes, the catalase, the carboxylases in their own little vesicles. And this is what the Golgi does. The Golgi takes a vesicle from the rough ER containing all this mishmash of proteins and sorts it out. And then at the other end of the Golgi, you end up with a vesicle containing nothing but the same protein. So we have the transport vesicle coming from the rough ER, that blue structure there, and then at the end of the Golgi, we have the vessel that's going to secrete, the secretory vesicle that's going to release the protein after it gets made. And the picture on the right is what it really looks like. That's 90,000 magnifications. So, now, and this is a nice little summary here. Proteins come off of the ER, the rough ER, in a vesicle, in a secretory vesicle, and they are transported to the Golgi apparatus, and the Golgi sorts everything out, and it gives the proteins three options. Get released from the cell, which is happening down here at the bottom, right there. We have, that's one approach. The other approach is to integrate those proteins with the cell membrane. And the third approach is to create digestive enzymes and store them in, or other organelles inside the cell. So we have these three options that can take place here. Now, some of these proteins are enzymes, and they're stored inside vesicles within the cell. One of these types of vesicles is called a peroxisome. Now, a peroxisome is designed to destroy what are known as free radicals. What's a free radical? Well, a free radical is a molecule or a part of a molecule that has a charge on it that's not right. Consider a sodium ion with a negative charge. Consider a water molecule with a charge on it at all. Consider a hydrogen ion. It's negative. So what happens here is these molecules become very reactive. They have the wrong charge on them, and they can cause lots of damage. As they move through a cell, they can destroy tissue. They can destroy organelles. As they move through tissues, they can destroy tissues, and free radicals can be associated with the development of cancer cells. And so we need an antioxidant to destroy these free radicals. So peroxisomes function in that category. Peroxisomes have two enzymes embedded in them. We want to get rid of the free radical. We want to destroy it. We want to oxidize it, and we want to prevent its we want to use an antioxidant to prevent the free radical from causing something to break down through oxidation. So we use oxidase to convert the free radical to hydrogen peroxide. Remember, hydrogen peroxide, H2O2. It's like water, only an extra oxygen. Oxidase, we convert the free radical, the toxin, if you will, to hydrogen peroxide. But hydrogen peroxide in the concentrations that the peroxisomes produce is dangerous. It's not like the stuff you find in the brown bottle down at Walgreens, for example. That's 3%. This is a lot higher concentration. So we have to break down the hydrogen peroxide, and we use catalase to do this, and that converts it to water. So we essentially burn it with, we oxidize it in the presence of oxygen, because you got to have oxygen for oxidation, and then but we form hydrogen peroxide by doing so, but then we can break that down using catalase. And so we destroy these free radicals. Now, another type, I'll just introduce this now and then we'll call it a day. Lysosomes are the other type of enzyme containing sac. They contain hydrolysis, enzymes that are, these are used to break down just about anything. Cell fragments, worn out cells, bacteria that might try to invade the cell, viruses which might try to invade the cell, organelles, as I said, that wear out. They can go after toxic substances, viruses, bacteria, funguses, protozoans, anything. And they will break down this by using these very powerful enzymes. They also are used by a type of bone cell to break down bone, to release the calcium stored in the bone. Now, when a cell becomes injured, the cell uses its own lysosomes to eat itself, to digest itself. It's called autolysis. So anyway, that's where we'll stop. We've covered a lot of ground today. Let me get out of this, and it's 10:58. Anybody have any questions before I close this up for today? Okay, then I'm going to get us out of here, and I'll see you all later on.